Hello, my dear students. Welcome you all to Sri Jain the Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to start with a very interesting topic under the section of biomolecules, proteins and amino acids. Proteins, the word is derived from a Greek word that means holding first place, right? It means what? Holding first place. The term was said by Berzelius and Mulder. So protein is derived from the Greek word which means holding first place, okay? And the term was given by Berzelius and Mulder. What are the general characteristics of proteins? Proteins, they are polymers. They are polymers of amino acids. And that is why they are also known as what? Polyamides, right? So proteins, the general characteristics of proteins, carbon, hydrogen, oxygen, nitrogen, they are the essential elements of proteins. Many proteins, they also have sulfur, okay, like the structural based proteins, which help to make the structures, to make different types of, exoskeleton. They are containing sulfur as a main ingredient. In some proteins, iodine is also there, iron is also there, phosphorus is also there. So these all inorganic materials, they act as a cofactor. They act as a cofactor. Okay. Proteins, they are the second most abundant compounds present in protoplasm. 7% to 14% amount of proteins approximately. So proteins, they are the second most abundant compounds present in the protoplasm, and their concentration is around 7% to 14%. Proteins, they are the polymers of amino acids given by Fischer and Hofmeister. So amino acids, the term amino acid was given by Fischer and Hofmeister. There are around 300 amino acids that exist. However, only 20 types of amino acids which are used in making proteins. When there are around 300 amino acids that exist, but only 20 amino acids they are used in making of proteins. Amino acid, they get polymerized to form proteins. The word amino acid was given by Hofmeister and Fischer. All the amino acids are amphoteric in nature, that is, they have two ends. One is the acidic and the other one is basic. Okay, so the acidic is the carboxylic group C double bond OH, and the alkaline is the amino group, that is NH2. Okay, so this is the acidic group and this is the basic group, amino group, and this is the carboxylic group. Understood? Now coming to next, this is the amino acid structure. You can see they are organic acids. Amino acids, they are organic acids. Organic acids. Okay. Now you can see this group is the most prior group, carboxylic group, and the group is attached to the carbon. This carbon is the alpha carbon. Okay. And the alpha carbon is also having the NH2 or amino group. Okay. So on the alpha carbon, they have a NH group, NH2 group, and a carboxylic group. Okay. Now, carboxylic group, it provides the acidic property to the amino acid, while amino group provides the basic reaction. Okay. That is why they are known as what? Alpha amino acids. Because the alpha carbon is having amino group, that is why these alpha on alpha carbon, they have amino group. To such type of carboxylic acids, they are known as what? Alpha amino acids. Understood? Now the alpha carbon is having a side chain, the alkyl group. Okay. Amino acids are therefore substituted methanes, where the four substituent groups occupy the four valency positions. So we can say that they are substituted methanes. as they have four different, you know, one is bonded to COH, one is bonded to NH2, one is bonded to H, one is bonded to alkyl group. Now coming to next, the R group, it changes, and because of the change in the R group, the classification of amino acids or the group of or of the family of amino acids is created. Okay, not only the change of R group, it depends upon the various pointers. R is represented as by H in glycine. Like for example, if we instead of R we take H, so R is equal to H in glycine. When R is equal to a methyl group, it becomes alanine. Okay, so R is changing, it becomes hydroxymethyl group, hydroxymethyl, it becomes serine. Okay, so this is the structure of amino acid. You can see, except glycine, except glycine, all alpha carbon containing amino acids are asymmetric. Except glycine, all amino acids are asymmetric. That is, they have a chiral carbon. Means they will show optical isomerism, optical rotation. Mostly all amino acids are levorotatory. Minus or small l. They are levorotatory. There are some non-protein amino acids which are present in antibiotics and all. The non-protein amino acids, they are dextrorotatory. Okay. This is a picture which will help you to understand the 20 common amino acids. Out of these 20 common amino acids, total 20, out of these 20 common amino acids, 7 amino acids are essential. 2 amino acids are semi-essential. And there is one more amino acid which is needed by adult. One more amino acid. Okay. So now, so out of 20 amino acids, these 20 amino acids, 7 amino acids are at most necessary, mean they are essential and they are present in all the organisms of all the age groups. Okay, 20 amino acids, especially when we talk about humans. So for humans, the 20 amino acids, out of 20 amino acids, 7 amino acids are essential. Okay, and 2 amino acids, they are needed by children's. Okay, and one of the amino acid is needed by adult. So these 2 amino acids which are needed by children, they are known as what? The semi-essential amino acids. Okay. Now, this is the classification. Alanine. Okay. Alanine, the letter which is coded for alanine, or which represents alanine, is ALA. Okay. And here's a genetic code which is given. This genetic code which will code for alanine. Okay. So alanine, you can see, is a aliphatic amino acid. Okay. Glycine, GLY, it is also a aliphatic amino acid. Isoleucine. Isoleucine, small I, capital I, sorry, or ILE. It is represented as ILE. Okay. It is a essential amino acid. Okay. While alanine and glycine, they are non-essential amino acids. So isoleucine is a essential amino acid. I will write down E. Okay. I'll change the ink. Yeah. E. Then leucine, it is also a essential amino acid. E. Okay. Leucine. Coded by capital L, okay, or LEU, leucine, LEU. Isoleucine, ILE, glycine, GLY, ALA for alanine. Then proline, non-essential amino acid, proline, it is a non-essential amino acid, aliphatic. Valine, it is also an essential amino acid, okay, VAL, valine. Then phenylalanine, it is a aromatic amino acid and it is essential and aromatic. Okay, so phenylalanine is essential and aromatic. PHE. Tryptophan, essential and aromatic. Tryptophan. Tyrosine, it is non-essential but aromatic. Tyrosine is non-essential but aromatic. TYR. Aspartic acid, it is non-essential and it is acidic. It is non-essential and it is acidic. Okay. This color will code for acidic. The aspartic acid is acidic. Already acid is given in the name as well. ASP. Glutamic acid, it is also acidic. GLU. Arginine, it is basic. ARG. Non-essential, 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 non-essential. Tyrosine, aspartic acid, glutamic acid, arginine, non-essential. Okay. Histidine, essential. Okay. HIS. Lysine, essential. Okay. Basic. Lysine is basic. Histidine is basic. Arginine is basic. Okay. Serine, it is having a OH group or alcoholic amino acid. It is having a OH group or alcoholic amino acid. It is non-essential. Okay. Serine, hydroxylic group. Threonine, it is essential and this is needed by adults. Threonine, okay, adults. Then cysteine, non-essential. Methionine, essential. And methionine is sulfur containing. Cysteine also sulfur containing. Okay. Serine and threonine OH group containing. Cysteine and methionine sulfur containing. Out of which methionine is essential and threonine is essential. Asparagine. Asparagine is amidic. Okay. It is having a amidic linkage. Glutamine, it is also having a amidic linkage. So amidic linkage is present in asparagine and glutamine. Understood? Okay. So I will go through again. Essential amino acid: isoleucine, leucine, valine, phenylalanine, tryptophan, histidine, lysine, threonine, and methionine. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So out of these 9, many books they haven't mentioned histidine as essential amino acid, but histidine is a essential amino acid. Okay, and see, methionine, threonine, lysine, histidine, tryptophan, phenylalanine, valine, leucine, and isoleucine. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So these 9 amino acids are essential. Okay, so many books they haven't mentioned or they have mentioned. So being on safe side, you also mention that 9 amino acids they are essential. 9 amino acids are essential, okay, and 2 amino acids are semi-essential, okay, and there is one amino acid which is needed by adult. Understood? Is that clear? Okay. Then sulfur containing amino acids cysteine and methionine, amidic containing amino acid asparagine and glutamine, then OH group containing serine and threonine, okay, then basic amino acids arginine, histidine and lysine, acidic amino acid glutamic acid, aspartic acid, aromatic amino acid phenylalanine, tryptophan, tyrosine. So phenylalanine, tryptophan, tyrosine, they are aromatic amino acids. Aliphatic amino acids, alanine, glycine, and aliphatic. So just out of three, these three all are aliphatic. Okay. It is mentioned selenocysteine is often referred as the 21st amino acid, but it is encoded in a special manner. In some cases, to distinguish between asparagine and aspartic acid, glutamine and glutamic acid is difficult. In these cases, code ASX, and it is not for your use. Okay. These are heterocyclic amino acids, hydroxyproline and proline. Okay. Now, important fact that proline, it gets metabolized to form hydroxyproline. Okay, like lysine, it metabolized to form hydroxylysine. So these hydroxyproline and hydroxylysine, they are known as rare amino acids. Rare amino acids. Okay, so these are rare. They have a heterocyclic ring. You can see the NH group amide linkage. So instead of amino group, they have NH group, amide linkage. That is why they are known as what? Heterocyclic amino acids. Understood? So these rare amino acids, they are non-coded amino acids. Rare amino acids, they are non-coded amino acids. They are non-coded amino acids. These rare amino acids, they are derived from the coded ones, okay, but with the help of modification. Okay, later on I will come. Now, the abbreviation of first three letters is used for the naming as I have shown you in the previous slide. Okay. Although there are many changes for amino acid derivatives like, asparagine is a amide. Okay. Like asparagine is a amide. And the abbreviation used is ASN, not ASP. Okay. So in the previous slide, you can see that the first three letters they almost are used, okay, but just a isoleucine ILE is used, okay, leucine LEU, proline PRO, BAL, PHE TRY is not used, TRP is used, TYR, then aspartic acid ASP, GLU, like that. Okay, the first three letters are used for the nomenclature of amino acids. Okay, now so I've explained all the types like neutral amino acids. So neutral, those who are not acidic and basic. Acidic, you remember glutamic acid and aspartic acid. Okay. Basic are arginine, histidine, and lysine. Apart from these, all are what? Like glycine, alanine, valine, leucine, isoleucine. They all are neutral amino acids. Okay. They all are neutral amino acids. I'll write here neutral amino acids. Glycine, alanine, valine, leucine, and isoleucine. Okay, GLY, ALA, VAL, LEU, ILE. Okay, so these all are neutral amino acids. Okay, neutral amino acids. These amino acids, they have only one amino group and one carboxylic group, mean they are monoamino monocarboxylic acid, and they don't have any cyclic hydrocarbon chain. Okay, they don't have any cyclic hydrocarbon chain. Now, out of these 21 amino acids, selenocysteine is under the research and observatory. Now you can see the charged and uncharged amino acids or the polar or non-polar amino acids. Okay. Arginine, histidine, and lysine, AHL. They have polar and charged side chains. Polar and charged side chains. Understood? Now, and positively charged. Polar and negatively charged side chains are in aspartic acid and glutamic acid. Negatively charged side chains. Okay. Then amino acid with polar uncharged side chains, serine, threonine, asparagine, and glutamine. Okay. So you have to remember AHL, AHL, arginine, histidine, and lysine. They have polar and positively charged. Glutamic acid, aspartic acid, negatively charged. Okay. Then uncharged, they are polar but uncharged. Polar but uncharged. It includes serine, threonine, asparagine, glutamine. सब तुम्हारा सब तुम्हारा आओगे कहाँ सब तुम्हारा आओगे कहाँ ओके serine threonine asparagine and glutamine so serine threonine asparagine glutamine they are polar and they are uncharged now there are some special cases like cysteine, selenocysteine, glycine, proline, special cases. Okay, cysteine, selenocysteine, glycine, and proline. Okay, as now positively charged are alanine, histidine, and lysine. Okay, negatively charged aspartic acid, glutamic acid. Polar but uncharged. सब तुम्हारा आओगे गांव. Cysteine, sorry, serine, threonine, asparagine, and glutamine. Okay, special cases cysteine, selenocysteine, proline, and glycine. Amino acids with hydrophobic side chain non-polar. They are non-polar. Alanine, isoleucine, leucine, methionine, phenylalanine, tryptophan, tyrosine, valine. Okay. So you have to remember. I will tell you a good trick that you remember these like positively charged AHL, arginine, histidine, lysine, negatively charged aspartic acid, glutamic acid. Then polar but uncharged सब तुम्हारा आओगे गांव like serine, threonine, then asparagine and glutamine. Okay. Then special cases cysteine, selenocysteine, proline and glycine. Apart from that, all are non-polar. Leucine, isoleucine, tryptophan, tyrosine, valine, methionine, all are non-polar. There are free amino acids present in the protoplasm as ions at isoelectric point. So isoelectric point, it is basically a point, or it is a pH of a particular solution at which the free amino acids they are present in protoplasm in ionic form. In ionic form. Isoelectric point is the pH point at which amino acids are stable in the electric field. Okay, and they can exist freely. They can exist freely. 10 amino acids from total 20 are not synthesized, and hence they are obtained from diet or food. Out of 10, 9 are essential and 2 are semi-essential. Okay. These depending amino acids are called essential amino acids like threonine, valine, lysine. Okay. Phenylalanine, tryptophan, leucine, isoleucine, methionine, arginine, histidine, where arginine and histidine are semi-essential. Arginine and histidine are semi-essential. Okay. Are semi-essential. Okay. 10 amino acids are synthesized in the animal body, which are called non-essential amino acids. Okay. Then glycine, proline, alanine, aspartic acid, glutamic acid, serine, glutamic, again it is named, cysteine, asparagine, and tyrosine. They are non-essential amino acids. Like glycine, proline, alanine, aspartic acid, glutamic acid, serine, cysteine, asparagine, and tyrosine. Okay. In the next class, I will be telling a good mnemonic to remember all these names, both for essential and non-essential. Eukaryotic proteins, they have L conformation, okay, amino acid, while bacteria and antibodies, they have D conformation amino acid. Capital D and capital L means what? See, they are the relative configurations, okay. So the eukaryotic proteins, they have L conformation amino acid, while the bacteria and the antibodies, they have D conformation amino acid. Amino acids are linked with the peptide bond to form protein. They are linked to peptide bond, okay, to form protein. Peptidyl transferase enzyme catalyzes the synthesis of peptide bond. Peptidyl transferase enzyme synthesizes the peptide bond, okay. Property of protein depends on sequence of amino acid and configuration of protein molecules. Property of protein depends upon the sequence of amino acid, okay, and configuration of protein molecules. L conformation of amino acid, I'll tell you. See, this is the alpha carbon. If the NH2 group, like this is alanine, in D conformation or L conformation, okay, L conformation, a particular group lies to a left hand side, and in D conformation, a particular group lies to the right hand side. Okay. So suppose we consider NH2 along with COH. If NH2 in comparison to COH or methyl group, it lies to on the left side, then it is L conformation. Understood? L conformation. Okay. Amino acids, they are linked with peptide bond to form protein. Peptidyl transferase enzyme catalyzes the synthesis of peptide bond. Property of protein depends upon the sequence of amino acid and configuration of protein molecules. Rare and non-protein amino acids, these are the uncommon amino acids. Non-coded amino acids, non-coded. In addition to 20 amino acids, proteins may contain uncommon amino acids which are derived from common or standard amino acids like hydroxyproline from proline. They are found in collagen. Hydroxylysine from lysine. They are also found in collagen. Okay, so they are modified form. Okay, so they are rare amino acids. Gamma carboxyglutamate, which is found in blood clotting protein prothrombin. Okay, gamma carboxyglutamate, which is found in the blood clotting protein, that is prothrombin. So they are rare and rare amino acids, non-coded amino acids. Okay. Now, when we talk about the non-protein amino acids, there are many amino acids which live in both free state as well as in the combined state, but not in the form of protein. So, non-protein amino acids means They live freely in cytoplasm or in combined state, but never in the form of proteins. But never in the form of proteins. But they never exist in the form of proteins. Many of them they are found in antibiotics and are called amino acid analogs. They are the part of non-protein amino acids. Amino acids. They are present in antibiotics and they make amino acid analogs. Other non-protein amino acids take part in important biosynthetic pathways like ornithine and citrulline. Ornithine and citrulline. Ornithine and citrulline, they are involved in urea cycle, Krebs and slit cycle, are involved in urea cycle. So they are also non-protein amino acid but involved in a metabolic reaction. Okay. Then homoarginine and diaminobutyric acid is a source of nitrogen. Homoarginine and diaminobutyric acid are the source of nitrogen. Source of nitrogen. Gamma aminobutyric acid is an inhibitory neurotransmitter. All these are non-protein amino acids. Non-protein amino acids. Okay. Gamma aminobutyric acid. Okay. All examples of non-protein amino acids. Except written in the box. Okay. Now, coming to next. Zwitterine. Zwitterine is basically a property of amino acid, especially when we talk about the isoelectric point or isoelectric pH. So zwitterine, you can see at a particular pH at an isoelectric point, amino acid molecule exists in a ionic form freely in the protoplasm or cytoplasm, in which the carboxylic group act as a proton donor and amino group as a proton acceptor. That is why this molecule is known as what? Amphibolic or amphipathic molecule. Okay. When you can see at high pH, when on decreasing the protons, on decreasing the protons, the basic character increases, and in at high basic character the COOH releases its proton, okay, and it exists in this form. Okay, this is at high pH, anionic form. At cationic form, when the H plus is added, okay, or proton are added, then it forms this form, cationic form, NH3 plus. The basic group gets activated. So at low pH, the basic group's activated. At high pH, the acidic group's activated. Okay. This is the zwitterion in which the ionic form of amino acid can be seen. Okay. At a particular pH. Okay. The pH which shows the zwitterion. Okay. That is known as what? Isoelectric point or isoelectric pH. Then essential amino acids. Essential amino acids, I know. In case of animals, all the amino acids which are required for the protein formation are not synthesized. Okay. Plants, they can synthesize all the essential amino acids. Plants, they can synthesize all amino acids they required. But animals, they cannot synthesize all amino acids. So amino acids which are not synthesized by animals, we get from diet or plants. Okay. The essential amino acids which our bodies cannot make, when we talk about the humans, the essential amino acid which our body cannot make: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So these are 9 essential amino acids plus 2 semi-essential amino acids which are needed by children. That is arginine, okay, and histidine. So histidine is counted as a semi-essential amino acid, okay, histidine and arginine. So isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Okay. So these are the amino acids which are required by us very much to make proteins. Okay. But they are not synthesized by us. So that is why we get from diet. Okay. Understood? Peptide formation. A peptide bond is a special type of amide linkage formed between two molecules where an alpha carboxyl group of one molecule reacts with alpha amino group of another molecule, releasing a water molecule. Okay. The peptide bond is also referred to as a isopeptide bond, where the amide bond forms between the carboxyl group of one amide amino acid and amino group of other amino acid at other positions than at alpha. Then it is known as what? Isopeptide bond. The process of formation of peptide bond is an example of condensation reaction, as uh, it involves the removal of simple materials like water. So it is a condensation reaction or a dehydration reaction. Peptide bonds are covalent bonds that exist between any two amino acids, resulting in a peptide chain. A partial double bond exists between carbon and nitrogen of the amide bond, which stabilizes the peptide bond. A partial double bond exists between carbon and nitrogen of the amide bond, which stabilizes the peptide bond. And that is why carbon, this bond is not movable. It is not under the rotation. It cannot move. Okay. And as it cannot move, this partial double bond exists, okay, between carbon and nitrogen. And as it cannot move, but our side chain and other groups can move. Okay. It also show resonance as the lone pair of nitrogen donates to carbon. Okay. So this resonance actually make the carbon nitrogen double bond stable. Okay, and molecule also it provides us stability. So peptide formation it takes place when the alpha carboxylic group of one amino acid combines with the alpha amino group of the other amino acid. Okay, so the alpha carboxylic group of one amino acid is known as what? Carboxy terminal. And the amino group, free amino group is known as what? Amino terminal or N terminal. Okay. The peptide bond is also known as isopeptide when the carboxylic group of one of the alpha carbon combines with non-alpha carbon, mean amino group of non-alpha carbon. It can be beta, it can be gamma, it can be delta, any. Peptide bond formation is a condensation reaction which involves the removal of water, mean it results in the dehydration. This you can see peptide and peptide bond, amino acid 1, amino acid 2. This is N terminus, free NH2 group. This is C terminus. The C terminus combines with the N terminus of the other group, okay, to form H2O. The H2O is removed. So C combines with nitrogen, and this C combines with nitrogen with a partial double bond character, okay. Carbon oxygen is having double bond, but this double bond can be shifted here as well. Carbon nitrogen double bond can also come because of the lone pair of nitrogen, like this. I'll show you. Okay, I'll show you. C double bond N H H R2 double bond OH. Right now, this C double bond C carbon H and H2. Now this comes to this side and the lone pair comes to this side. So a partial double bond character can make the bond strong. Understood? Yeah. So this is dipeptide. When two amino acids are linked, then it forms oligopeptide. If some more, amino acids are linked like that, and then polypeptide. Okay. Functions of amino acids. Functions of amino acids, these are the building blocks of proteins and enzymes. They store the nitrogen content for recycling nitrogen. Glycine helps in the synthesis of protoporphyrin, that is heme, okay, in blood. Tryptophan, it helps to synthesize indole acetic acid, auxin hormone, okay, and nicotinamide. They are energy carriers. Tyrosine, it helps in the formation or synthesis of thyroxine, melanin, and adrenaline. Okay. Histamine, uh, histidine, sorry, it is not histamine. Okay. Basically, histidine give rise to histamine. It is derived from, I'll write this way. Okay. Histamine, no, it is a potent vasodilator and a bronchoconstrictor. Okay. It is derived from histidine through decarboxylation. Okay. Histamine is required for optimum functioning of muscles. Optimum functioning of muscles. Okay. There are some amino acids which act as a donor of methyl group like methionine. donor of methyl group. Okay. Ornithine and citrulline are the components of urea cycle. We have already done that. Okay. Then there are many amino acids which are the part of antibiotics. Okay. Enkephalins, they are the pentapeptides produced by nerve cells. Pentapeptides produced by nerve cells. So, my dear students, it is almost done. We have completed the amino acids and the general concentration, general features, general characteristics, and the types of amino acids. In the next class, we will be starting with the proteins, their types, their organization, their functions. Okay. Till then, everyone, please take care of yourself. Bye bye. Take care. Stay safe.